Hello, Bio32ers. Professor Newton here again. We are looking at muscles 2 today. As you can see on page 79 in your lecture notes, this is going to be physiology of skeletal muscle fibers, what I also like to call the physiology of the neuromuscular junction. What I decided to do with this lecture is split into two parts, part 1 and part 2. Part 1 is where I want to discuss the electrical signal we call an action potential. Then I want you to take a break. I want you to learn those steps and then come back for part 2. Part 2 is where we'll see how that electrical signal produces a mechanical outcome. So with that said, let's get started. As we know, skeletal muscle fibers and muscles themselves cannot contract on their own. They've got to be told when to contract by a neuron. There's a special type of neuron that stimulates skeletal muscle fibers, and those are called somatic motor neurons, or neurons of the somatic nervous system. If you hear that word somatic, you want to think voluntary. That's the voluntary branch of the nervous system. What we find is that the cell bodies and the dendrites of these somatic motor neurons are located up here in the brain and spinal cord of the nervous system. What they then do is they fire their electrical signals called action potentials down their axons that get bundled together with nerves that then stimulate the skeletal muscle fibers. You may wonder why we're using an electrical signal. That's because there are only two types of signals in the body, electrical and chemical. Because sometimes we have to travel over a great distance or also muscle contraction and needs to be almost instantaneous, we want to use the fastest signal possible, and that is our electrical signal. Before we talk about how we generate that electrical signal, I want to do a little housekeeping. And I'll do this all the way through this entire Muscle 2 lecture. What I've done is I've gone back in the lecture notes and made sure that everything that I say orally is also listed in your notes. So with that intro, let's go back to page 79 and let's look at an overview of nerve stimulus and action potentials. By way of overview, the nerve cells that activate skeletal muscle fibers, as I just said, are called somatic motor neurons, or neurons of the voluntary branch of the nervous system. Their cell bodies reside in the brain spinal cord, and their long thread-like extensions called axons, bundled within nerves, transmit electrical signals that we call action potentials to the muscle cells that they serve to initiate contraction. Well, now that we know that, let's talk about how we generate this electrical signal we call an action potential. The first thing we want to look at is we want to look at a cell at rest. The cell at rest here in front of you is the motor neuron. The same thing applies here to our skeletal muscle fiber. What we can do is we can let a little box here represent the skeletal muscle fiber or the motor neuron. The inside of our box will represent the intracellular environment. The outside of our box will represent the extracellular environment. And again, it's exactly the same for the skeletal muscle fiber as well as the motor neuron. As we've learned before, the cell membrane separates again the inside from the outside, producing two different environments. There are a lot of differences between the intracellular and extracellular environment. One of the differences we want to focus on today is charge. What we say in physiology when there's an electrical charge, we call that a potential. When it's across the membrane, we call that a membrane potential. When the cell is at rest, we call that a resting membrane potential, and a resting membrane potential just simply implies what I just said, an electrical charge across the membrane when the cell is at rest. When the cell is at rest, what we find is that the inside of the cell is negatively charged with respect to the outside. That's why you see all these little negative signs. The inside is negative, the outside is positive. If we were to put an electrode in there, we see that it's about 70 times more negative. There are many reasons for this, such as negatively charged side chains of amino acids and many more, but what we're going to do is focus on just two important reasons. Two important ions, one is sodium and one is potassium. Looking at a real cell here, this is what that looks like. Again, we've inserted an electrode here, in this case in the axon of the somatic motor neuron, and we see the inside is about 70 times more negative. We say the resting membrane potential is negative 70 millivolts. Again, a lot of reasons for that. Let's focus on this first one. The first reason we want to look at is this ion here that we now know as sodium. I draw the sodium ion in a particular way for a reason. Number one, I draw it in rather large, so this shows that there's a lot of it. I also use these brackets, and you'll remember from our discussion in chemistry that the brackets refer to concentration. So what I've got here, though, in this drawing is I've got a large concentration of sodium outside the cell. I draw it exactly the opposite when it comes to inside the cell. Inside the cell, you can see that I draw it rather small. It shows that there's a small concentration of sodium inside the cell. When it comes to potassium, what we have is exactly the opposite. We have a high concentration inside the cell and a low concentration outside the cell. And then I have my sodium potassium pump over here that is used to establish and maintain those concentration gradients. You actually may remember this from our discussion on cells. When we talked about membrane transport, I actually used this as an example, told you that we'd eventually get to it. Well, today is that day. As I look at the diagram on the screen before you, what we see again are these concentration gradients. Due to the selective permeability properties of the membrane, those ions cannot freely move. That's because they're charged. So as a result, what we have is basically like a charged battery ready to do work. In order to generate the signal that we call an action potential, what we're going to do is we're going to move these ions. The only way to move these ions, however, because they cannot freely diffuse through the membrane, are through channels. So we're going to employ channels in order to make that happen. Before we talk about those channels, again, let's do a little housekeeping. Here, point B on page 79, let's talk about membrane potentials. The resting membrane potential. The nerve cell and muscle cell are like a charged battery ready to do work. Due to the selective permeability properties of the membrane, ions cannot freely cross and are therefore unable to reach equilibrium. Also, due to the active transport by the sodium-potassium pumps, as you can see in the diagram before you, pumps 3 sodium ions out for every 2 potassium ions in at the cost of ATP, we establish and maintain a concentration gradient with, again, a high concentration of sodium outside the cell, low concentration inside. Potassium is the opposite, high concentration inside, low concentration outside. The net result of this is an intracellular environment that is more negative than the extracellular environment, again, approximately 70 times more, a resting membrane potential of about negative 70 millivolts. Note, there are other ions and factors that contribute to the resting membrane potential that we're not going to discuss at this time. Top of page 80, the resting membrane potential can also vary anywhere from negative 40 millivolts to negative 95 millivolts depending on the type of cell. But we're going to simplify our discussion by always using negative 70 millivolts as our resting membrane potential for the purposes of our class. So with this charged battery that's ready to do work, how do we move these ions? As I referred to before, again, they cannot move on their own. So what we're going to do is we're going to use channels. There are 2 types of channels that we'll use, what we call ligand-gated channels and voltage-gated channels. What you see on the screne before you is an example of a ligand-gated channel. A ligand, what that is a chemical messenger. If that chemical messenger, that ligand, is released by a neuron, we call that a neurotransmitter. If the ligand or neurotransmitter is present and it binds, the channel opens, ions can move. If the neurotransmitter or the ligand is not present or it does not bind, the channel stays closed and the ions cannot move. So ligand-gated channels, they open and close in response to the presence or absence of a ligand. Where we find ligand-gated channels are the dendrites in the cell body of the motor neuron, and also at what we call the neuromuscular junction or the motor end plate here of our motor neuron, where this white squiggly line is. The second type of channel we find are voltage-gated channels. Voltage-gated channels open and close in response to a voltage. We get to a certain voltage, the channel opens, the ions can move. We get to another voltage, the channel closes, the ions cannot move. So again, where ligand-gated channels open and close in response to the presence or absence of a ligand, voltage-gated channels, what they do is they open and close in response to a change in voltage. Whereas the ligand-gated channels, again, were found on the motor end plate here of the skeletal muscle fiber, also the cell body and the dendrites of our motor neuron. The voltage-gated channels are found down the axon of our motor neuron and also the sarcolemma here, the membrane of the skeletal muscle fiber. By the way, there are many more voltage-gated channels than there are ligand-gated channels, and they're also much faster. So again, doing a little housekeeping, let's look at graded potentials and local depolarizations, page 80. At point number 2a, the electrical signal or impulse called an axial potential can be defined as a reversal in the membrane potential. What we're saying here, folks, is that we're going to take the inside of the cell at rest that is negative and we're going to make it positive. That is the definition of our axial potential, a reversal in the membrane potential. The way we're going to do that is through the movement of ions. There are 2 types of channels that make this happen. As we just said, our ligand-gated channels and our voltage-gated channels. The ligand-gated channels open and close due to the presence or absence of a chemical signal called a ligand, more specifically a neurotransmitter that binds the receptor site. The ligand binds, the channel opens. The ligand unbinds or is not present, the channel closes. Again, as we said, localized to the dendrites in the cell body of the neuron, as well as the neuromuscular junction motor end plate of the skeletal muscle fiber. The voltage-gated channels open and close in response to a change in voltage. The membrane potential reaches a certain voltage or charge, the channel opens. The membrane potential reaches a different voltage or charge, the channel closes. These are widely distributed along the axons of neurons and the sarcolemma of the muscle fiber, and they act much faster compared to the ligand-gated channels. So how do we produce this electrical signal called an action potential? When it comes to looking at an action potential, the easiest way to understand it is simply to graph it. What we're going to do is we're going to graph the change in membrane potential. As we just defined, an action potential is a reversal in the membrane potential. So what we're going to see is basically we go from negative to positive. Once we hit that positive membrane potential, that is going to be our action potential. As you can see on the screen in front of you, we've got time on the x-axis, and look at that time. It's in milliseconds. That's less than a second. That's how fast these action potentials travel. On the y-axis, we've got millivolts. Let's go ahead and throw in a couple important charges here. As we look at these charges, some important ones today will be negative 70 millivolts, negative 65, and also positive 30. Putting that all together, that's what an action potential looks like. We start at negative, we go positive, and then we go back negative, and then we're ready to fire another action potential. So let's talk about the steps to produce this important electrical signal that we call the action potential. So to get this started, what happens is we have to start way up in the brain and spinal cord. You remember from a second ago, again, the cell body and the dendrites are way up in the brain and spinal cord of the nervous system. Well, these somatic motor neurons that you see on the screen in front of you, they're controlled by other neurons in the nervous system. And what happens is those other neurons release their own neurotransmitters. When that occurs, those neurotransmitters are going to bind onto receptor sites on the dendrites in the cell body of this motor neuron. When we talk about those receptor sites, what we're actually talking about are ligand-gated sodium channels. So when the neurotransmitter binds, the ligand-gated sodium channels open. Because we have a high concentration of sodium outside the cell and a low concentration inside the cell, sodium will start to diffuse in. As it starts to diffuse in, sometimes we say it starts to leak in, what happens, it carries positive charge with it. When it carries that positive charge, we get a stepwise movement right here to a more positive value. The membrane potential moves from negative 70 to negative 69. We call that a local depolarization. When we use that term depolarization today, polar means different, depolarization means less different. As we move from negative 70 to negative 69, we become less different. The inside of the cell is becoming more positive, so it's becoming less different, or again, more like the outside. It's starting to become more positive. Meanwhile, there are more ligands binding onto the receptor sites on the cell body and the dendrites of this motor neuron. Again, those receptor sites are actually ligand-gated sodium channels. By the way, the name tells us what the channel actually is doing. Again, we'll open and close only in response to the presence or absence of that ligand, and only sodium is allowed to move. But as we bind more and more ligand-gated sodium channels, more and more ligand-gated sodium channels will open up. Sodium having a high concentration outside, low concentration inside. Sodium will continue to leak in, producing more and more local depolarizations. The membrane potential will then move from negative 70 to negative 69, negative 68 to negative 67, and so on. If we get enough ligands binding to the receptor sites to open up enough ligand-gated sodium channels to get enough sodium to leak in to produce enough local depolarizations to hit negative 65 millivolts, we fire. We will then hit threshold. Threshold is defined as the minimum amount of depolarization required to initiate an action potential. What does that mean? It's the minimum amount of movement toward more positive value. Negative 65 is more positive than negative 70. The minimum amount of depolarization or movement toward more positive value that's required to initiate the action potential. The action potential itself is what we say is all or none. That means if we hit threshold, we fire. We get the whole graph. If we don't hit threshold, we don't fire. If we get to negative 65.5, are we going to fire? No, we have not hit threshold yet. If we get to negative 65.4, still not good enough. Negative 65.2, we're still not going to fire. Negative 65.1, we're not going to fire. But if we get to negative 65.0, we will fire. Again, it's all or none. If we hit threshold, we get all of it. If we do not hit threshold, we do not. So once we hit threshold, that is the signal to open up the voltage-gated sodium channels. When the voltage-gated sodium channels open, we have high concentration of sodium outside the cell, low concentration inside the cell. What we'll do is we'll get a massive influx of sodium. As sodium rushes into the cell, it floods the cell. Because sodium is positive and because we have so many voltage-gated sodium channels and they work so fast, we are literally flooding the cell with a positive charge. As we flood the cell with a positive charge, the membrane potential skyrockets here to a plus 30. We call this the depolarization phase. Again, the inside of the cell is now becoming less different than the outside. We started negative, now it's positive. It's plus 30, which is essentially the same as the outside. That's the basis of your action potential. Meanwhile, as we continue, once we reach plus 30, that is the signal for the voltage-gated sodium channels to close and the voltage-gated potassium channels to open. Potassium having a high concentration inside and a low concentration outside, what happens is potassium rushes out of the cell. As it rushes out of the cell, it's also carrying positive charge with it. Because we're carrying positive charge with it, we're losing positive charge to the outside of the cell. The membrane potential plummets down back to a negative 70 millivolts. We call that the repolarization phase. Once we hit negative 70 millivolts, what happens next is that's the signal for the voltage-gated potassium channels to close and the sodium-potassium pumps reestablish their gradients. At the conclusion of the action potential, you see this little dip right here. We call this dip, though, the refractory period. That's a short time after the action potential fires where we cannot fire another signal. That's because all those concentration gradients are out of whack. We've got a ton of sodium on the inside, a ton of potassium on the outside. So it's just a short period of time where the sodium potassium pumps go to work and they reestablish their concentration gradients so that we can fire. Meanwhile, what you've done is you've initiated an action potential right here at the top of the axon. What will happen is that action potential will propagate. Propagate means to spread. Like knocking over a stack of dominoes, that action potential, though, as long as we hit that first domino, will propagate down the axon of the motor neuron, or also the sarcolemma here of the muscle fiber, and that's what will be used to initiate contraction in the skeletal muscle fiber. Meanwhile, again, let's do a little housekeeping. Still here on page 80, let's drop down to the bottom, point D. Reviewing the steps that we just discussed, let's look at them in the notes. When the cell receives a signal, that means that a neurotransmitter is bound its receptor, the ligand-gated sodium channel is open, and sodium begins to leak into the cell down its concentration gradient. This produces a stepwise progression toward a more positive charge, and we call this a local depolarization. The membrane potential then moves from negative 70 to negative 69, top of page 81, and so on. As more and more ligands bind, more and more ligand-gated sodium channels open, and more sodium leaks into the cell, causing further local depolarization. This then continues until we reach threshold. Threshold is approximately negative 65 millivolts. It can be defined as we just defined it as the minimum amount of depolarization, that is to say, the minimum amount of movement toward more positive charge that is required to initiate an action potential. Once again, it is all or none. That means that if threshold is achieved, an action potential is fired. If it's not, no action potential takes place and the cell returns back to its resting membrane potential. Note, if threshold is achieved, then no more ligand neurotransmitter is necessary and the voltage-gated channels take over. We are now done with the ligand-gated channels. They're technically done at this point. Meanwhile, to our steps of the action potential. The action potential is defined as a reversal of the membrane potential with a total change in voltage of about 100 millivolts as the membrane potential skyrockets from about negative 70 to plus 30. Two phases, again lasting less than a second. The depolarization phase. At negative 65 millivolts and once threshold is achieved, the voltage-gated sodium channel is open. Sodium rushes into the cell down its concentration gradient, produces a huge influx of sodium, and this produces a rapid depolarization. The membrane potential skyrockets here to a plus 30. Then the repolarization phase. At plus 30, that's the signal for the voltage-gated sodium channels to close and the voltage-gated potassium channels to open. Potassium rushes out of the cell down its concentration gradient. We say that produces a huge efflux of potassium, and this produces a rapid repolarization. The membrane potential returns down to a negative voltage. Top of the next page in your lecture notes, page 82. This then continues until the membrane potential reaches a negative 70 millivolts, at which time the voltage-gated sodium channels close and the sodium-potassium pumps reestablish the concentration gradients. As we just said, we've got the refractory period, defined as a short period less than 1 second after an action potential has occurred where the cell cannot immediately fire another signal. This is the direct result of the temporary destruction of those concentration gradients that we discussed earlier in response to the movement of the ions. So the sodium-potassium pumps must reestablish the concentration gradients before another signal can be fired. Meanwhile, propagation. In nerve cells and muscle cells, as an action potential is reached on small sections of a cell's membrane, they set off other sections of the membrane to pass threshold. It is self-propagating. The action potential propagates only in one direction, away from the origin of the nerve impulse, much like a domino effect. Again, in the case of the motor neuron, down the axon of the motor neuron, or in the case of the skeletal muscle fiber, down its sarcolemma. Meanwhile, the net result is the nerve cells release neurotransmitters at their presynaptic or axon terminals, or muscle cells contract. And that's where we're going to take it with our next step. So with that said, what I've done for you here is I've outlined each of the major events from our notes in our discussion today as far as what's happened in each voltage. As you can see, everything we've talked about is outlined here as well as your lecture notes. You also can use your textbook. Your textbook will discuss this as well, but again, as usual, do not worry about any of the details that we did not discuss. It doesn't happen as a threshold. It uses -95 instead of -70, but you get the idea. What I'd like you to do is to, again, take these steps. Take, again, the drawings, though, that I produced for you here as part of this presentation. Try to recreate those on your own. What I want you to do is then to memorize that. I want you to be able to draw the picture of the actual potential from memory, and I want you to be able to list each and every one of these steps. If you can at least do that before you rejoin me, though, for part 2, where we pick up here at the neuromuscular junction, then you're going to be in good shape moving forward. As usual, if you have any questions, please don't hesitate to reach out. Otherwise, that's all I've got for you for part 1. I'll be joining you shortly for part 2 whenever you're ready. Thanks so much, guys.